Sickle cell disease, an inherited blood disorder that affects red blood cells, is a menacing health challenge faced by millions of people worldwide. Characterized by abnormal, crescent-shaped cells that block blood flow in blood vessels, this disease causes a cascade of health problems such as severe pain, organ damage, and even stroke. According to the World Health Organization, sickle cell disease is a significant public health concern, contributing to a mortality rate of approximately 5% in children under the age of 5 in sub-Saharan Africa. With these alarming statistics in mind, have you ever wondered how gene editing can revolutionize the treatment of diseases like sickle cell? Our journey starts the discovery of DNA in the mid-20th century, a fundamental building block of life. This was the first time scientists could peek into the blueprint that makes us who we are. As we delve deeper the world of DNA, we began to understand that changes or mutations in this blueprint can cause genetic diseases. These are conditions passed down from generation to generation, affecting millions of individuals worldwide. This revelation was like finding a needle in a haystack. A tiny alteration in the vast expanse of our genetic code could lead to debilitating illnesses. The next logical step in this journey was to find a way to fix these mutations. This quest led to the birth of techniques. The concept is simple yet revolutionary. If we can precisely change the faulty parts of our DNA, we could potentially cure genetic diseases at their source. In the late 20th century, initial gene editing techniques were developed. However, these were often imprecise and inefficient, akin to trying to edit a book with a sledgehammer instead of a pen, but they set the foundation for what was to come. Fast forward to the early 21st century, and we've seen a quantum leap in our ability to edit genes. This progress is largely thanks to a revolutionary tool known as Crispr, a game-changer in the field of genetics. In essence, gene editing can be likened to a spell-check tool for our DNA. It can find the faulty spelling or mutations in our genes and correct them. This has the potential to revolutionize the treatment of genetic diseases, transforming them from incurable to potentially curable. This understanding of our genetic makeup set the stage for the development of innovative gene editing therapies. Enter Crispr, a revolutionary tool that brought gene editing to the forefront of medical research. Standing for clustered, regularly interspaced short palindromic repeats, Crispr is a system that scientists borrowed from bacteria. Yes, you heard it right, bacteria. These microscopic organisms use Crispr as a kind of immune system to fend off viruses. But how does it work in the realm of gene editing? Picture this. Crispr is like a pair of molecular scissors. Guided by a molecule known as RNA, it can cut DNA at precise locations. This allows scientists to alter, remove, or replace specific genes. And the beauty of it is in its precision and efficiency. Before Crispr, gene editing was like trying to edit a book with a blindfold on. With Crispr, scientists can now locate and edit a single word in that book with ease. The first applications of Crispr were carried out in lab settings, on cells in a petri dish. Scientists were able to prove that they could successfully edit genes, but the real challenge was applying this in living organisms. However, the potential to treat genetic diseases was clear. Imagine being able to correct a single genetic error that causes a disease like sickle cell anemia, or being able to turn off the genes that contribute to the development of certain cancers. The possibilities seemed endless. The journey of Crispr has not been without its hurdles. Like any new technology, it faced skepticism and a myriad of ethical and safety concerns. However, with each hurdle crossed, the potential of this tool became more and more evident. With Crispr, the dream of curing genetic diseases was no longer a distant fantasy, but a tangible reality. As we continue to refine this technology and understand its implications, we stand at the cusp of a new era in medicine, an era where we might finally have the tools to rewrite our genetic destinies. But how did we go from lab experiments to FDA-approved treatments? Well, the journey from lab to clinic is a strenuous one, filled with rigorous testing and meticulous validation. It all starts with preclinical studies, where the safety and efficacy of a potential therapy are evaluated in laboratory settings. These studies involve testing on cells in a dish, and often on animals, to understand the biological effects of the therapy. The next stage is clinical trials, a multi-phase process involving human volunteers. Phase 1 trials are primarily concerned with safety, determining the dosage and identifying side effects. Phase 2 trials examine the treatment's efficacy while continuing to monitor safety. Phase 3 trials compare the new treatment with the current standard treatment in a larger group of people. Now let's focus on the journey of the first gene-editing therapies for sickle cell disease that recently received FDA approval. These therapies, Casvi and LGenia, went through a rigorous approval process. They were developed by Vertex Pharmaceuticals and Bluebird Bio respectively, and they utilize groundbreaking Crispr technology and gene therapy. The approval came after extensive clinical trials that demonstrated these treatments' potential to significantly improve the quality of life for sickle cell patients. The trials showed that patients receiving these therapies experienced fewer painful episodes and blockages, with the most significant concern being off-target editing and the location of the gene insertion for healthy hemoglobin. However, it wasn't all smooth sailing. The journey to approval was marked by challenges. For instance, Bluebird Bio faced multiple hurdles, including a pause in trials and an extended review period for its therapies. Despite these setbacks, the company persevered, leading to the eventual FDA approval of LGenia. The approval of these therapies didn't mark the end of the journey, though. Both companies are required to follow patients for 15 years to monitor any potential long-term effects, showcasing the FDA's commitment to ensuring the safety and efficacy of these groundbreaking treatments. This approval marks a significant milestone in the history of medical science. This is not just a victory for the companies involved, but for the entire medical community, and most importantly, for patients suffering from sickle cell disease. The journey to FDA approval is a testament to the power of persistence, innovation, and rigorous scientific investigation. So, what are these revolutionary therapies that have received the FDA's green light? Let's start with the first one, Casvi. This therapy, developed by Vertex Pharmaceuticals, uses the groundbreaking Crispr gene editing tool. Here's how it works. The therapy involves transplanting modified blood stem cells back into the patient. These altered cells boost the production of fetal hemoglobin, which in turn prevents the sickling of red blood cells. The result? A significant reduction in the blockages that cause the pain and complications associated with sickle cell disease. Then we have LGenia, the brainchild of Bluebird Bio. Like Casvi, LGenia aims to increase the population of healthy blood cells in patients. But here's where it differs. The primary concern with LGenia is where the gene for healthy hemoglobin is inserted, while with Casvi, the worry is about off-target editing. Now let's talk about potential benefits. Studies reviewed by the FDA showed that patients who received either of these therapies experienced fewer painful episodes and blockages. This is a significant stride forward in improving the quality of life for those living with sickle cell disease. But let's not forget about the risks. The most significant concern with these therapies is long-term effects. That's why the FDA has asked both Vertex Pharmaceuticals and Bluebird Bio to monitor their patients for 15 years after treatment. And lastly, we need to address the elephant in the room, the cost. These therapies come with a hefty price tag. Casvi could exceed $2.2 million per patient, and LGenia comes in even higher at $3.1 million. This is a critical factor that could limit access to these potentially life-changing treatments. These therapies are not just treatments, but potential cures, offering hope to thousands of sickle cell patients. What does this mean for the future of gene editing therapies? This is a question that has been on everyone's minds since the recent FDA approval of the first 2 gene editing therapies for sickle cell disease. The implications are significant and far-reaching. The approval of these therapies is a clear testament to the potential of gene editing technologies, like CRISPR, to transform the landscape of medicine. It opens the door for the development and approval of more gene editing therapies for other genetic conditions. From cystic fibrosis to Huntington's disease, the possibilities are vast and exciting. Ongoing research in this field is robust and promising. Scientists are continuously improving the precision, efficiency, and safety of these gene editing tools. They are also exploring the use of gene editing to treat complex diseases like cancer, heart disease, and neurological disorders. However, the road to a future where genetic diseases can be effectively treated or even cured with gene editing is not without challenges. There are ethical considerations that need to be addressed. For instance, where do we draw the line between therapy and enhancement? How do we ensure the equitable access to these potentially life-saving treatments? Technological limitations also exist. Off-target editing and long-term effects of these therapies are concerns that require further research and monitoring. The approval of these therapies comes with a 15-year follow-up requirement to monitor any potential long-term effects, indicating the cautious approach being taken. Finally, there's the issue of cost. With price tags reaching into the millions per patient, how do we make these therapies affordable and accessible to those who need them the most? These are complex issues that require thoughtful discussion, continued research, and innovative solutions. But the approval of these therapies for sickle cell disease has set a precedent. It's a testament to what can be achieved when science, innovation, and regulatory bodies come together with a common goal: to improve and save lives. While there is still much work to be done, the approval of these therapies is a promising step towards a future where genetic diseases can be effectively treated or even cured with gene editing. However, as we move forward into this new era of gene editing therapies, there are challenges and ethical considerations that we must confront. The line between therapy and enhancement is a delicate one. How do we ensure that these technologies are used responsibly and ethically? And as we push the boundaries of what is medically possible, we must also consider issues of access and affordability. These therapies have the potential to save lives, but their high cost can put them out of reach for many. We must work toward solutions that make these life-changing treatments accessible to all who need them. Despite these challenges, the potential of gene editing therapies is undeniable. As we continue to refine these technologies and navigate the ethical landscape, we stand on the cusp of a new era in medicine, an era where genetic diseases that once seemed untreatable are now within our grasp to cure. This is the power and promise of gene editing. Closing. Thank you for watching. If you found this video informative, please give it a thumbs up and don't forget to subscribe for more content like this. As we continue to explore the exciting world of gene editing, we invite you to join us on this journey. Together, we can unravel the complexities of this revolutionary technology and its potential to transform medicine as we know it. Until next time, stay curious and keep learning.